just to get started, so I'm going to talk about imaging data as well, and to orient you, we have our gray matter and we have our white matter, and those are sort of very broad divisions that oversimplify things, but our gray matter is kind of our processing in our brain, so it's what processes information, and then our white matter are, as Dr. Taylor had said, are the communication highways. So we do our processing in local regions, but it's really the white matter, so these sort of connections here, that connect distant brain regions and allow for information processing across regions in so-called neural networks. And that's really critical for sort of high-order, complex functioning. And actually, our gray matter and our white matter are continuous. So we can see here that what makes gray matter look gray are all the cell bodies, the neurons, and then the white matter, the reason why it appears white is because there's a fatty layer around it, and that allows for this sort of saltatory conduction, if you remember from science in high school, this fast transmission of signals. And fast transmission of signals between distant regions is really what underlies really complex functioning again. And we can image this in a really cool way. So we can track movement of water molecules in the brain using a type of MRI imaging, which all the families who are involved in brain imaging do as part of the imaging protocol. And the movement of water molecules is very different in white matter as opposed to gray matter. So if you think of cables with lots and lots of wires inside, water molecules are not going to be able to go across. They have to go along, just like water along a straw. And so you get a very different signal from areas of the brain that house white matter as opposed to other areas of the brain. And we can capitalize on that difference of signal and extract information indirectly about the structure of underlying white matter. And so when we do imaging in this way, when we're in white matter regions, we get this kind of cigar shape, which gives us information about how water molecules are moving in white matter regions. And we can actually reconstruct the three-dimensional trajectories of white matter tracks using the information from each voxel, or kind of pixel, of our pictures of the brain. And this is one method of doing this, but you actually just take information from neighboring pixels that have similar types of information, and you can reconstruct the trajectory of the white matter track, and you end up with these beautiful, beautiful pictures. And this is actually why I ended up doing imaging, because I love learning about networks, and this was the first time that I was ever able to see this three-dimensional aspects of these networks. So you can use this information to recreate the networks of the brain. And these are some different networks. This is a network that's involved in different kinds of things, but one is sort of face processing. The cingulum bundle is, some people say, is the seat of emotional functioning. It's a very central big tract that integrates most of the cortical aspects of the brain. And so people have done really amazing things with using DTI information, and I'm going to tell you one story that's not a pond story, but I'll tell you a good pond story in a second. So this is a study where they looked at high-risk siblings, so siblings of children who had a diagnosis of autism, which is what made them high risk for developing autism themselves. And they actually tracked infants. So they imaged infants at 6 months, at 12 months, and at 24 months. And at 24 months, some of those infants had gone on to a diagnosis of autism. So they could separate the groups as to people who remained high risk but didn't have a diagnosis, and people who ended up having a diagnosis, or toddlers, I should say. And so these are a number of different white matter tracks that they looked at. And this is the power of longitudinal imaging, so imaging at multiple time points, is that you get to see what changes. So at 6 months, in red are the kids who had a diagnosis of autism, and they have white matter structure that looks different from the high-risk siblings. They actually look like they're developing a little bit faster. And then if you look at 12 months, there's no difference. And at 24 months, their white matter actually has changed trajectories completely so that it looks like it's developing slower. And rather than going through all the details, that's kind of the take-home message. But if you were to just look at the 12-month time point, you wouldn't see any difference between the groups. And this is really why longitudinal imaging is so powerful, because you can see this trajectory. So this is why when, if you get asked to come back, this is what makes that contribution so important, to be able to track that trajectory. Because a different story is the story of, and we've heard it already this morning a little bit, sort of 10 years, so we did a review of 10 years of diffusion tensor imaging. So this is that MRI type of imaging that's important to look at white matter. And we looked at it because connectivity is thought to be very, very important, not just in autism, but in lots of different developmental disorders that affect children and adolescents, like ADHD, like OCD. And so this review of DTI studies, which actually largely consisted of studies that had small samples, so like 20 to 30 kids in each group, and also comparing kids with a disorder or autism and kids without a disorder. So using this kind of case control approach. And there were widespread differences found across a lot of the studies, but as you've heard already this morning, the differences didn't align very well. So really heterogeneous findings. A lot of differences, but in different spots. And some of the stories suggested that if you look at different age groups, you're going to get different findings. And a lot of our data comes from very wide age groups, sort of looking at kids from 7 all the way up to 17, where there's a lot of developmental change going on in those kids. So it brings up these issues that maybe, autism is a very heterogeneous condition. Different kids with the condition look very different. But we probably have to resolve some issues in terms of wide age ranges. We need bigger samples. We need longitudinal data. And so we need new models, right? And so the PON model is a really unique one where we now have data from 3 different types of disorders that affect children. So we just finished a study where we looked at 71 kids with autism, 31 kids with ADHD, and 36 kids with OCD, and we compared them to kids without a disorder. And what we found was, one, there were some similarities across the kids. So this image shows you that just in this little part of the brain, part of the corpus callosum, which is a huge tract that connects the left and right hemispheres and is really important for things like cognition. There were differences across the groups. So the same direction in that area, we found slight differences in the integrity or the structure of that region, and it was the same. So decreased in autism and ADHD and OCD. This was the first time, compared to controls, this was the first time that anybody's ever really looked across these groups and published on it. So then the next thing that we found that was really interesting was, regardless of what your diagnosis was, the structure of your white matter in, I don't know if you can see it very well, in little red, yellow spots on these white matter maps, associated with your everyday functioning. So the better your everyday functioning, the better your white matter looked, regardless of what your diagnosis was. The worse your everyday functioning, the worse your white matter looked. And so we've been kind of talking a little bit about how do we translate findings in brain imaging and neuroscience into the clinic. And one of the things that we're trying to look for is biomarkers, markers of difference that you can actually go after with a treatment. So the significance of this study is it's the first time a study has found similarities in brain wiring in children with ASD, ADHD, and OCD. No prior study has been carried out in this way yet. Similar changes in wiring suggest that some of the same treatments may have relevance across disorders. And then identifying particular wiring linked to problems with function provides a basis to work towards developing treatments that may improve function and may secondarily improve wiring. So this is the goal. So bringing neuroscience into the clinic, using the advances that we make in brain science, and bringing new treatments to improve function and outcome in individuals. And so the hope is that one day, somebody gets a diagnosis, but then they also get a profile of what are the real difficulties that they're having in their everyday life. And with these everyday difficulties, regardless of what their diagnosis is, are there certain treatments that could actually target those differences? And we know that it could improve outcome, and we know that it actually changes the issue that is there to begin with, whether it's a brain imaging finding or a cognitive finding or a genetic finding. And maybe it's a behavioral intervention, maybe it's a medication treatment, maybe it's something like brain stimulation, which we're actually working on another study. But the issue is, you need the biomarker, you need the target to go after, and then you need to work to develop new treatments. So just give you a little plug for a study that we're doing now at CAMH, where we know that kids with autism, young people and adults, have difficulties with executive functioning. So executive functioning are problems with organization, task initiation, task completion. And in that transition to adulthood, when you have to become independent, executive functioning is really critical for all of us in terms of how well we do in life. And so we actually know what the brain circuit is that's involved in executive functioning. And we can go after that circuit with something called brain stimulation. And this is something that we've been doing a little bit of in other disorders, in people who have depression and people who have schizophrenia, and there's some really nice positive early results. So we're undertaking a study, and we have been for the last couple of years, where people come in and they do brain imaging before the study, then they do 4 weeks of brain stimulation treatment, and then they do brain imaging again. And they're randomized to either get the treatment or get sort of a placebo type of the treatment. And the idea is that you embed imaging, you use imaging in the clinical trial to see, if you can change something, how are you changing it? What is the mechanism of the improvement in behavior? How is that related to changes in the brain? So how do we move forward towards the goal of bringing neuroscience into the clinic? So the question is, what if you had unlimited resources, what do you need? And we really need participants. We need brain imaging studies that have a lot of participants. And the reason is because these disorders are very heterogeneous, and there's probably not one finding there. We need big groups in order to get the signal. We also need to pair treatment studies with efforts to better understand how treatment works, and we need to follow people over time. So the whole push for longitudinal imaging is probably going to be a very important one in terms of translating to the clinic. So thank you all very much for being here today and for being part of POND, and there's a number of people.
Okay. When you talk about brain stimulation, that 2 slides back,
yeah.
How is that done?
Right.
What is the nature of stimulation?
Yeah, so we use something called a transcranial magnetic stimulation. It uses a magnetic coil that allows for a transfer of an electric current into the brain, and this is what we're using. Do you have a follow-up question to that, or no? That's it. Yeah. Other hands. Yeah, up there.
I have a question. It's not necessarily specific to neuroimaging, but how much closer is POM and other organizations like POM to medicalizing the diagnosis of ASD? That's just like ASD.
You mean to finding a biological underpinning for a diagnosis?
Parents know the big thing we realize is your child's diagnosis is not a medical diagnosis, it's a mental health diagnosis largely. And therefore, the services you get are different depending on largely the functionality or how hard you find it. But children who have a medical diagnosis, that's a different kettle of fish.
Well, the tricky thing is you've asked a psychiatrist that question. So I would flip it on its head, and I think that the main issue is that we don't see mental health diagnoses as medical diagnoses. That we need to destigmatize our diagnoses, and I think part of the effort and actually, being amidst the discussion in psychiatry and real efforts to destigmatize mental health is that we are starting more to think about function, right? How can we get people back to what it is that they want to do in order to move forward with their lives, and rather than, the stigma of what it is and whether it's medical or mental health. I mean, the brain it's as medical as you can get, right? So that's my answer to that question.
This is a very important discussion and a very difficult discussion.
Yeah.
And in fact, we have a parent panel in the afternoon, so we should talk more about it. But I want to put my neurologist hat, right? So these are cultural constructs, right? We make cultural decisions about what we call a disease and what we not call a disease, and what we consider stigmatizing and what not. So in the case of mental health, when we talk about disorders, people think it's stigmatizing, but I can tell you when you have a kid with epilepsy and you say a child has epilepsy, nobody thinks that's stigmatizing per se. And it's partly because we do understand the biology, and we are very clear that if you have asthma, you have a problem with reactive airways, and if you have epilepsy, you have a problem with too much excitation in your brain. And when it comes down to neurodevelopmental disorders we don't understand very well, we all struggle with where to put them. That's one problem. The second problem is that these disorders exist on a spectrum of function, like Stephanie said. So there are plenty of people who carry some of these traits. They have this biological difference, but don't experience function and definitely do not want to be seen as having a disorder. And that is an experience that needs to be honored also. So it has become a very difficult conversation. The downside of not medicalizing is exactly what you said. Early intervention for autism is not covered under the Canadian Health Act. Why? Because this is not considered biological treatment. Although we all know better, this is as much of a biological treatment as anything else, right? So there are certain aspects of our healthcare system that currently are not meeting our needs when we don't admit that we have a brain-based difference, right? On the other hand, when we medicalize too much the experience of people who do not see themselves as having a disorder, we miss the kind of uniqueness and the strengths that come from having those types of differences. And in fact, we know those types of differences are extremely important in society. They have been important for years. People who are detail-oriented and very focused to take the autism side of things and not particularly getting distracted with social clues have been extremely contributory to society. People with ADHD, I would like to believe, given I have a little bit, is that are the creative people who have the big, they're not very good at putting things in place, but they have the bigger ideas about how things should be done and what needs to be done. So it's a hard balance. I can tell you that from PON's point of view, we are committed to finding the biomarkers. We're committing to figuring out what the biology that produces all these differences are. We're also committed to make sure that the way this gets communicated to policy, to funding agencies, and so on, reflects not only the healthcare needs of the kids who actually need healthcare, kids and adolescents and adults, but also the perspectives of the people who feel that this particular difference has been meaningful to them and to those around them and contributes to society. So hard discussion, excellent question. We should probably get back to it during the parent panel. Maybe we'll go ahead, you pick.
Go up there, yeah?
For studying, we have multiple children. One has ASD, ADHD, and ODD. Our other one is LP and ADD. Now, the study, is it strictly just for ASD, or are LP and ODD, or ADD children able to study also?
Sorry, so is the question that is the PON study only for those kids who have one diagnosis? No, it's not. So kids are primarily identified as having one primary diagnosis, but you can have multiple diagnoses. And if we limit to people who just have one diagnosis, we're excluding most of the people who have these types of difficulties, because as you can see, the brain we don't have one area of the brain where autism lives and one area of the brain where LD lives. It overlaps quite a bit. And so a lot of the people that are in the data that I presented have multiple disorders.
So we just contact you at that website to get involved with this?
Yeah.
Absolutely. Yes.
Yeah? Okay.
Yes, go ahead, right there.
What's the result post-treatment? Or is it still being done?
No, we're not there yet. We're almost there. But this is where the push comes. So, if anybody knows somebody who's between the ages of 16 to 35 and experiences executive function difficulties, we're really trying to finish up recruitment for this study so we can get to the answer of whether it helps.